Seminar to tell you a little bit about nutrients and why I'm fascinated by them and why they're really important. And you know, I also thought, because you like German speakers, I don't know how many German speakers you've already had this semester, but I'm probably the third or fourth one. But anyway, this will be a very, you know, it gives a global overview, but then I'll also talk a fair bit about Queensland and specifically about sugarcane. But I want you to know more about nutrients in case you've missed some of these things in your school education, or you're not working on nutrients because there are obviously so many other things to work on. But the key thing is that crop productivity has increased at least fourfold throughout the world since about the 1950s. And of course, we've had lots and lots of changes because of improved varieties, improved agronomy, you know, chemicals and pests and pathogen control, but also fertilizer. And this is sort of graph that you see here, sort of fairly typical one for many crops. This is corn in the US from 1860 to 2010, and you can see there's this distinct increase somewhere in the mid-20th century where the yields are going up. And at that time, it was not just the improved varieties that were coming on board, there was also an increasing use of fertilizers, and that's sort of my topic today. Now, this is the good news. Of course, we have improved crop productivity, and we need to improve crop productivity, obviously. But there's also another fact that's not so good, and that's the pollution of nutrient. This paper was a sort of global overview that was published a few years ago, and it just sort of summarized what a lot of people were feeling or realizing what was going on. So we have areas, boundaries, safe boundaries here indicated in green of the planet, and 3 of them have been surpassed: the biodiversity loss, climate change, and then the other one that may be a surprise to you is the nitrogen cycle. So nitrogen has increased. We have enriched our biosphere with nitrogen, with reactive nitrogen, and that is considered by these authors as, you know, a hugely surpassed boundary. So that's going to be a major part of my talk, but I'll also link it into various parts of agriculture, as you'll see along. Going back in time to the sort of start of the 20th century, there were 2 people, Fritz Haber and Carl Bosch, who invented what you probably all have heard about, the Haber-Bosch process. And the Haber-Bosch process allows the use of air dinitrogen and converting it into reactive nitrogen. And over the course of the 20th century, it started around about 1910, and then there was a steep increase here in the consumption, or the production and consumption of reactive nitrogen. And we're now at about 147 megatons of nitrogen that is available and mostly used as for fertilizer. In green here is the world population, and again, you know, we're about at 7.2 billion people today, and you can see both of them track very closely. Now, explosives, of course, was the initial reason why Haber and Bosch were asked by the German government to come up with this way of making reactive nitrogen, because the Germans were gearing up for the First World War, and then later on, of course, for the Second World War, unfortunately. But very early on, already in 1914, Bosch set up experimental sites to study the synthetic nitrogen, this new type of man-made nitrogen for agriculture. But to put this in perspective with today, or over the course of the 20th century, this recent paper from China showed that they achieved a 3.4-fold increase in food production over the 50 years from the 1960s, but they achieved it with a 37-fold increase in nitrogen fertilizer application. So in other words, you had to put on 10 times more nitrogen in order to achieve a tenth of that increase in agricultural productivity. And that, of course, is at the core of the problem of this nitrogen pollution that is, you know, really jarring the benefits of crop production. So, but before I go on and talk about pollution and what to do about it, I want to talk a little bit about where do fertilizers come from, because it's a really interesting story that, you know, goes into, again, the global realm and you know, how nations developed and what happened. So initially, very early on, since about the late 19th century, people mined sodium nitrate, Chile saltpeter as it was also called, or the white gold, in the Atacama Desert. And that's the sort of picture of it. And it was a very strange geological formation that accumulated nitrate and sodium. And that was export. You can see the production was mostly Chile in 1913 and then Germany and a few other nations. But that was, and of course at the turn of into the 20th century, the global shipping of nitrogen, Chile saltpeter, was sort of getting a bit unreliable, and that's why the Germans started to think of alternatives. There was a second source of fertilizer as well, and that's still being in use today, which is also called the Inca's gift of the gods, the rich nitrogen source and phosphorus source that the seabirds have deposited along the Peruvian, Chilean coast of South America. Here, very nutrient-rich ocean waters full of seabirds, and these seabirds land and deposit their guano in these mountains of bird droppings. And you can see people who are mining it. These are the actual birds that deposit it, and then here, this is today's, where people are still hacking away with primitive hand tools and brushes and brooms to remove this precious nutrient for agriculture. And again, that's something which really shaped the history of South America, the British, Chile, Peru. There's a big historic history in this as well. But even in Queensland, there was nitrogen and phosphorus fertilizer. The first income of the Queensland Treasury was through guano mining on Lady Elliot Island in the Great Barrier Reef. This is a picture here of Lady Elliot. And if you've ever been there, you will notice there's very little native vegetation left. That is because it got scraped off the guano and the vegetation was removed. Other Great Barrier Reef islands are in much better shape. But it's still ongoing. Countries like Nauru have basically been completely extracted. So this is all rocks, coral, and guano, and the guano is being mined as an agricultural nutrient. And of course, this is now the nitrogen that is manufactured from air dye nitrogen. So we have unlimited amounts of air dye nitrogen, and we can make ammonium nitrate and urea. This is what urea is the most commonly used form today in agriculture. Tiny little pellets dissolves very quickly, and then here you can see rice farmers throwing on the urea onto their crop. Up the top left corner is a picture of this first urea factory in Oppau in Germany, where they're geared up from very low levels to very high amounts of nitrogen, mostly in the form of ammonium and nitrate early on, and very, very quickly over a couple of decades. You might see this big crater here in the foreground. This is actually after a huge explosion in 1921 where about 600 people got killed, and they could hear the bang of this explosion 300 kilometers away in Munich. So it killed a lot of people, many got injured, and they realized that the way they were storing the ammonium nitrate wasn't such a great idea. But there was another type of bang in the biosphere. So this reactive nitrogen that has nearly tripled in the past century is now getting to very high levels. So I'm just talking you through this figure here, sorry. We have nitrogen-fixing crops, we have fossil fuel, we have industrial nitrogen like nylon and other substances, and we have nitrogen fertilizer. And you can see the fertilizer is going up and up and up throughout the 50 or so years. And then we also have the range of what's called natural nitrogen fixation or biological fixation. I'll talk a little bit more later on. And you see the range is there's a big span. And the big span is because we're not very sure how much nitrogen is actually fixed by organisms, by prokaryotic organisms in natural ecosystems, symbiotically or not symbiotically, because it's actually very hard to quantify. But in any case, what you can see is that we have enriched the Earth very dramatically with reactive nitrogen. And that's, in perspective, the carbon cycle that we worry about. We worry about elevated CO2. We worry about changes that we're doing to the planet's biogeochemistry. We have perturbed that by only 5%. So 300 versus 5, a huge difference. And people think that about almost half of the people on Earth are currently fed as a result of this ability to make synthetic nitrogen fertilizer. And Václav Smil, who's written this very enlightening book, Enriching the Earth, Fritz Haber, Karl Bosch, and the Transformation of the World Food Production, he calls it the biggest experiment ever in biogeoengineering. Now, I would challenge this statement, actually, and I will talk a little bit more about this, but I don't think we're completely and utterly relying on the synthetic fertilizer use and fixation. We need to look at other ways, and there's probably other ways that we can deal with this. But anyhow, that's how a lot of people see it at this point in time. This picture I wanted to show you, not just because obviously advertisement worked very well already early in the 20th century. This is a picture advertising ammonium sulfate as a fertilizer. And so obviously there's quite a different sort of beauty ideal of what a woman should look like in those days compared to what we might think of today. But apart from that, you can see with ammonium sulfate, everybody is happy. So the farmer is smiling, his wife is very happy, and without ammonium sulfate, life looks a bit dreadful. Not only are there no crops, but there's also rocks and other things. So clearly, people have been brainwashed for a long time, and that's just one of the many ways of of how fertilizer has been promoted. But let's have a look in the pollution side, and I'll come back to the agronomy and the plant side of things a bit later on. The global nitrogen enrichment that I already talked about has been trying to visualize on a global scale by Galloway and others, and you can see 1860, the deposition. This is here global, across the globe, the deposition of nitrogen, kilograms of nitrogen per hectare per year, and sort of very low. There's a few patches where there's more nitrogen, and then it ramps up dramatically over the next 100 or 30 years or so. And then forecast for 2050, you can see there are parts of the world where 50 kilograms of nitrogen were expected to come down by deposition of reactive nitrogen per hectare and year. However, this is a total underestimate, because already today in Beijing, for instance, we get 90 kilograms nitrogen per hectare per year deposited. So this is in wet and dry deposition as volatile as a particulate. Very large amounts. That's more that some farmers put on their fields. So in other words, you're fertilizing areas that don't necessarily need to be fertilized. In fact, it's a big health risk for people, amongst other things. So talking about risk, the other thing that springs to mind when you talk about nutrient pollution are greenhouse gases. That's something that's really ramped up in the people's knowledge and perception over the last decade or so. And the International Panel for Climate Change, the IPCC, has been tremendously active in this area, promoting the dangers and this message. So emissions from agriculture overall have increased 17% from 1990 to 2005. Methane and nitrous oxide account for 50% and 80% of anthropogenic production of those two gases, and nitrous oxide alone accounts now for more than 10% of global warming. So it's a hugely powerful greenhouse gas. And you see here in the different bars, in red is CO2, green is the methane, and blue is nitrous oxide. And I just draw your attention here to these two bars. So 1999-2004, in agriculture, we have the largest amount of methane emitted. Much of this is from ruminant and cow, what do you call them, cow emissions. And then the nitrous oxide, which is mostly from soils. And again, while those two bars don't look all that huge, if you look at the global warming potential, nitrous oxide is 300 times the global warming potential of carbon dioxide, and methane around about 30. So they are very, very powerful greenhouse gases indeed. And we need to be very careful when we, create conditions where these are readily produced from soils and from agricultural operations. Now, nitrous oxide emissions are very much strongly linked to synthetic nitrogen fertilizer use, and this is a picture from the FAO web pages, and you can see here, this is again on a country basis. The darker red, the more nitrous oxide emitted. So China, sort of very dark, and then India, Brazil, US, again very dark red, and parts of Europe as well. Australia doesn't look so bad by and large, because these are the emissions calculated per, over the entire area of the country. But we do have areas that have very high emissions, and I'll talk about them later on. Now, when everything fails, when we don't have science, we can bring in the celebrities, and they help. And in this case, just a few days ago, Leonardo DiCaprio attended a conference called Our Ocean that was in Washington, D.C., and held by the State Department of State of the United States. And of course, anything that's held in Africa and in Washington is, of course, of great relevance. But the good thing is that he sort of commented on things. He said, since my very first dive in the Great Barrier Reef in Australia 20 years ago, to the dive I got to do in the very same location just 2 years ago, I have witnessed environmental devastation in firsthand. What once had looked like an endless underwater utopia is now riddled with bleached corals and massive dead zones. So, that was a big statement, and it's been going around the media just in the last few days, because, of course, on one hand, it's brilliant that he draws attention to the devastation that's happening to the reef, but at the same time, Australians are very worried, of course, at the impact of such a statement on tourism. But we're, of course, here concerned with nutrient pollution. So how does Leonardo DiCaprio link to agriculture and nutrient pollution? Well, from the US EPA, the Environmental Protection Agency, they said over the past 20 years, anti-pollution laws have greatly reduced discharge of toxic substances, and this is mostly with point source, so, sewage treatment plants and others. But no comparable effort has been made to restrict the input of nitrogen and phosphorus from dispersed or non-point sources such as agriculture and urban runoff. And that is, a major drama. And this is the same thing in Australia, where although people have been aware of it and have thought about it, to actually regulate it and to really tackle the problem has been, a huge problem. And that is across the world. That's not just the US or not just Australia, but in many other parts as well. So what are we talking about? We're talking about the Great Barrier Reef. So according to Leonardo, and this is obviously backed up by science, 50% of the coral in the Great Barrier Reef are dead. So we're going from here, from a healthy underwater utopia with lots of species and tremendous diversity, to areas that are pretty much dead. Corals are bleached, algae are overgrowing them, and then the crown of thorns starfish are, eating whatever's left. And so it's now very clear, and there's no way that this isn't true, because people have shown these connections, even though they're still part of, the industry, part of, the population who denies these kind of connections. But the nutrient pollution of waterways has been linked to phytoplankton blooms, and these phytoplankton blooms are fostering high survival of crown of thorns starfish larvae, and then followed by the large-scale destruction of the Great Barrier Reef by these adult animals. So this is here a crown of thorns starfish, massive starfish that eat their way through these corals. And they're normal. I mean, it's natural to have some of those in the reef, but the larvae, every adult female produces 13 million larvae. So most, normally only a few of them survive. But with phytoplankton enriched waters, these larvae, the survival rate is much, much larger, and you get far bigger outbreaks of these crown of thorns starfish. So that's to show you what does it look like. So here we've got part of the Queensland coast from Maryborough going up to Rockhampton, and you can see this is a satellite image. Obviously, the colors are not quite exactly correct, but you can see here the red line indicates where the nutrient pollution extends into, from the coast into the Great Barrier Reef. So there's a plume, and there's a bunch of things that come in, suspended solids as well as nutrients. So 100 kilometers into the reef. Here are some of the reef islands that you can see. Lady Elliot Island is actually one of those little ones here. And so when we look at the land use, of course, immediately agriculture springs to mind. And in this case, this is now here part of the Mackay area in central Queensland, north central Queensland. And you can see sugarcane in green, horticulture in red, and the vast majority is indicated here as sugarcane. There's also some grazing land and others. But sugarcane has been linked to, these sort of events of, high nutrient efflux runoff from the land. And if we look at the sort of numbers, since European settlement, annual loads to the Great Barrier Reef has increased. Nitrogen, nearly 6 times higher, 80,000 tons per year. Phosphorus, nearly 9 times higher, 16,000 tons per year. Suspended solids, which is a different word for soil that you actually want to keep on the land. Suspended solids, nearly 5.5 times to 17 million tons per year, and photosynthesis, photosystem 2 inhibiting herbicides that obviously went there before European settlement are now 30 tons per hectare, 30 tons per year. So these are large numbers, nearly an order of magnitude, close to an order of magnitude for phosphorus, for instance, increase into these Great Barrier Reef waters. And if you look at, this is now from the Queensland government statement, main contributors to the Great Barrier Reef's coral loss are storms and cyclones, nearly 50%, outbreaks of crown of thorns, starfish, 42%, and then bleaching from ocean warming. So quite clearly, the crown of thorn linked to the nutrients are accounting for about half of the coral deaths. So something that is so significant that recent discussions between the sugar industry and conservationists, where people said, well, we can either have the reef or we can have the sugar cane. We can't really have both. And that's how things have been going in just in the last few years. And so has there been nothing done? Of course things have been done. People have been, that's not a new problem. People have been thinking and working on this for a long time, but ultimately there has been very little improvement in curbing, for instance, the nitrogen pollution to the GBR. So here nitrogen, this is again, just part of the Reef Plan And they sort of indicate what has been happened, and there's been some significant improvement here with nitrogen a 7% improvement, so that's not particularly good. And that's partly because, of course, there's an echo effect. Once the soil profile is loaded with nitrogen, it'll take a long time to actually see an improvement, even if the land has been, the land management has improved. And that has sort of many other implications. So the WH drinking water quality is sort of 50 milligrams nitrate per liter. We have quite a lot of bores in Queensland where this is reached. That's a high level of nitrate. And the same with groundwater quality in natural ecosystems. It's a very high level of phosphorus, increased by about an order of magnitude, similar to the actual waters in the Great Barrier Reef. So the Reef Plan targets, so this is again by the Queensland government, what they're aiming for is reducing the nitrogen load from 80,000 tonnes to 30, they want to reduce it by 30,000 tonnes, phosphorus by 15,000 tonnes, and this one, by 2.5 million or by 15 tonnes. So it's a huge aim in order to produce better water quality to improve the reef. How that's going to be achieved is the key question now. And I'm going to talk a little bit now about, what can be done in order to address this monster problem. Because we have 2,000 kilometers of coastal catchment in Queensland, so that's a large area. Definite point source, definitely over a large area. So what we need is high production, low pollution agriculture. And here are just the 6 top crops in the world, from maize to soybean. But the one I want to talk about is sugarcane. It's by far the largest crop in terms of total biomass removed. But it's also, and even though you might not always see it with food plants, because it's also used for biofuel production, obviously, and fibers and other things. But sugarcane, of course, is a big industry in Queensland, $2 billion revenue, and we have some of the best sugarcane in terms of yield, disease resistance. So the Australian sugarcane breeders and industry can be very proud of themselves on their achievements from that perspective. But the global efficiency of nitrogen fertilizer and sugarcane cropping is quite low. It's only about 50%, similar to other crops. And that is despite the fact that sugarcane is not a grain crop. So in other words, you're not removing a nitrogen-rich, protein-rich kernel from the field. You're removing sugar, which is carbon. And Australia is sort of in the middle here. So you see in some countries, like especially India or China, the rates of nitrogen fertilizer go up to 800 kilograms per hectare per year. And the amount that's actually removed from the field ranges roughly 50 to 80 kilograms per hectare. So generally, there's about twice as much fertilizer put on the fields as what the crops actually end up in the biomass. And so some years ago, we stepped back and we wanted to sort of understand from a plant perspective, from a soil perspective, from an agronomy perspective, how do all these things link? And so some of Nicole Robinson's work then shows that, for instance, if you go, November is generally where the fertilizer goes on, and you get a very big peak of soluble inorganic nitrogen, ammonium nitrate, in the soil solution that ceases in about February, and then is sort of quite low for the rest of the growing season. And simultaneous with this increase in inorganic nitrogen, you get an increase in the flux of nitrous oxide. So in other words, because we have such high levels of inorganic nitrogen, we get high levels of nitrous oxide emitted. The crop, on the other hand, keeps on taking up nitrogen later into the growing season. So there's obviously a mismatch here where the demand of the crop is quite low, but the availability of this very easily available and easily lost nitrogen is very high. And you can measure this. So for instance, that was done as part of a PhD thesis from Diane Allen, and she measured what nitrous oxide emissions were with different types, amounts, and applications of fertilizer. You can see here 200 kilos of fertilizer applied in one go or in 2 applications, so with a few weeks in between, I think 4 or 6 weeks in between, 100 or 50 plus 50, and you can see that the emissions are actually lower, of course, depending on, the less fertilizer, the less emissions, even though at 0 fertilizer, this was because the field had been fertilized in previous years, so there's a lot of nitrogen still available. But you can see definitely there's a big drop between, 200 or 100 plus 100. And disappointingly, however, the industry, a companion paper was written about this, and they talked about the fact that there was no improved yield when you split the fertilizer application rate, and therefore it wasn't recommended because it meant the farmer had to go out and drive twice over the field in order to apply the fertilizer. But in that paper didn't mention at all that there was a big improvement in the emissions of nitrous oxide, so much less nitrous oxide emission if you split the fertilizer. These emission factors, by the way, they're just a way that the IPCC and others calculate how much of the fertilizer is converted into nitrous oxide. And by and large, for sugarcane and other crops, the IPCC use it the 1% as emission factor. You can see that we've got far more. And in fact, there's in some instances up to 20% of the fertilizer is emitted as nitrous oxide in extreme situations. So we're really dealing with a big problem from cropping, not just sugarcane, but other crop systems as well. And again, so when you think about an agricultural crop system, I mean, as a more ecologically trained person, I'd like to think in a broader context. So I like to think, what is a forest, the original vegetation in this area versus the one that we've created, an agricultural field. So, you can look at your sugarcane or other crop system and your fertilizer and the different fluxes and pools in the soil and, your losses as greenhouse gases and your leaching mostly as nitrate, because nitrate is very mobile in soil. And then you can compare it to another system, like a forest system. See, so what exactly is different? Obviously, different plants, different management. We're not plowing this, we're not tilling it, we're not applying fertilizer. But, it kind of helps to give you a bit of a benchmark if you want. So Richard Bracken, who's been, part of working on soil biological processes, he did an experiment where he went to different sugarcane soils that were conventionally farmed or a new farming system that has a range of improvements and compared it to an adjacent forest. And you can see here, nitrous oxide emissions from January to September are very high. In all of those sugarcane farms, there's a bit of a difference maybe between the farming system, but these are semi-quantitative data only. But the key thing I want you to take home from this is see how the forest, there's virtually no nitrous oxide, very, very tiny emissions from the forest, close to nothing compared to the sugarcane. Interestingly, on the other hand, the forest soil is doing us a big favor. It's doing us an ecosystem service, so to speak, because it's sequestering methane. So these are now negative fluxes. So this is drawdown of methane into the forest soil. And you can see the sugarcane soil emits a little bit of methane. Sometimes it draws a bit out, but it's sort of plus minus zero mostly. Whereas in the forest, we're actually taking methane out of the atmosphere. And if you remember back, methane was one of the top 3 greenhouse gases, 30 times more potent than carbon dioxide. So in other words, forest soil is taking in greenhouse gases, whereas our agricultural soil is emitting greenhouse gases. And then you start thinking, so why is that? Obviously, the conditions are quite different, but you still could think, so why doesn't the sugarcane soil sequester methane? And I think, at that stage, or maybe a bit before, we were actually thinking, well, obviously that must be to do with the biology in the soil because these processes are driven by microbes. So immediately, you go from looking at a broader picture, you have to drill down and say, well, let's try to understand what's actually going on in the soil. So I want to spend a little bit of time explaining to you how we see, with a focus on nitrogen, those different processes and the role that microbes play. And to do that, I sort of have to first introduce a few little concepts. So if in a very broad term, you have a plant here, the plant dies or decays, and you have a large pool of soil organic nitrogen, and that's in the form of protein. So it's organic nitrogen, and it's about up to 10,000 kilograms per hectare in the soil as protein. That's just the stock of nitrogen that's in the soil. That's not necessarily available. Some of that is, but a lot of it is just part of the soil organic matter. These soil proteins can be broken down into smaller units, such as peptides or amino acids, and then they can also be further broken down into ammonium and nitrate. So, and these are all microbial processes. I should have put some microbes here as well. But this is the kind of different forms of nitrogen that we find. And there's a few others, amino sugars and other things. But that's the sort of big, the big sort of lineup. And inorganic, as I just explained, this here is also called mineral nitrogen, if you come across this term. And that's ammonium, NH4+, or nitrate, NO3-. And so if you open any textbook, people will tell you our plants use nitrate, and they might also use some ammonium. And that's why we supply urea as fertilizer in most instances, or we supply ammonium nitrate, depends. And then these pools get bigger because the urea is very soluble, gets converted to ammonium very quickly, and then gets nitrified. And so that's what the plants are using. But also, of course, coinciding with this is microbial activities that will then take some of this ammonium or some of this nitrate and convert it into nitrous oxide. So we're back at our nasty greenhouse gas. We also find that nitrate is very easily leached from soil because it has negative charge. Soil also has negative charge in many instances, so we are not having a very good resident time of nitrate in soil. And so there are very complex interactions, in other words, between the fertilizer, the microbes, the soil, the crop, to do with fluxes, to do with uptake and release, and driving the system that obviously in most farming systems, sugarcane included, is not particularly efficient. We also looked at a paradigm because, as I said before, nitrate is always crops up as, many papers that you read, many textbooks that you look at. Nitrate is the main source of nitrogen for crops. But I knew that there were other, and people had done studies that there were other, some plants that don't use nitrate very well. So we were curious whether sugarcane was good with using nitrate or not so good. And again, I just now want to sort of bring this all together. So I'm going to talk about nitrogen, sugarcane with nitrate and different types of genotypes and the physiology of nitrogen. I will talk about soil and microbes that I've already mentioned are a major part of the whole equation, and a little bit more about the agronomy of sugarcane to sort of show you how you can come from plant nutrition and nutrients, how you can end up in talking about and thinking about agronomy with lots of stepping stones along the way. So about 10 years ago, we were lucky enough to get involved in a sugarcane CRC and were able to study how sugarcane genotypes behave with different amounts of nitrogen. So a very simple idea was there must be genotypes that are more efficient, that need less nitrogen, and therefore would be beneficial to grow because you don't have to fertilize them quite so much. And this is some of the very early work that Nicole Robinson did and others with low nitrogen genotype here, grown with replete nitrogen. You can see with low nitrogen, this genotype doesn't look very good. So in glasshouse, obviously. And then the second genotype, however, was still very good looking with a replete nitrogen, and with the same low amount of nitrogen, this thing was actually looking quite good. So in other words, you get a sort of idea that there are genotypic differences, and you can see here biomass at low nitrogen supply and biomass at replete nitrogen supply. If you plot those two, you get a whole range of genotypes. This Q165 is one of the parental lines. You can see it's pretty good. It's the best actually at high nitrogen supply, but it's not the best at low nitrogen supply. There are others that are actually better, like this one here, which is good at, the best at low nitrogen supply, and is also pretty good at high nitrogen supply. So we thought there's a genetic reason, and obviously a way that molecular breeders can use that information to select better genotypes. Obviously, then you get the criticism going, yeah, it's all very nice, you do this in the glasshouse, but does this hold up in the field? So you go into the field and you do maybe fun things like get a blimp to take some remote sensing images, and that looks all very cool, and the students would flock if you offered them such a project. Reality is, so it's generally a fairly hard slog. This is our first harvest of just 7 genotypes, but sugarcane is a lot bigger than Arabidopsis or wheat or other things. So, but in the end, what we now have is 2 trio trials. The second one is being wrapped up this year, 2 hectares in Mackay and Ayr, where we tested 64 genotypes, and this is at the recommended nitrogen rate. So the darker red color means, very vigorous, and then at a lower rate, you can see there's a big difference. Each of these squares, I should explain, that is a genotype of sugarcane, so it's a replicated block experiment with each of the genotypes being tested multiple times to see how they perform in much lower nitrogen conditions. So 40 kilograms, again, is very, very low, but we wanted a low rate just to ensure that we see differences because of this big effect of soil residual nitrogen that will always be around in these kinds of experiments. And just some early data. This is sort of work that Nicole's wrapping up this year, and she will give a seminar once we have this story sort of complete. But you can see here, these are all the 64 genotypes lined up, and 20, 40, up to 60 kilograms nitrogen per hectare accumulated in the biomass, and only 20 kilograms was provided to the plants as fertilizer, so that took up more nitrogen, including from the soil. And you can see that, the genotypes, the poor performing one, like this one here, only took up 8 kilograms of nitrogen from the soil, whereas a very well performing one, like the one up the top here, could acquire 41 kilograms. So that's a difference of, more than 4-fold difference in an efficient and a not so efficient genotype. And then, of course, you can say, well, but what is the nitrogen that the plant acquires? I said, does it translate into different biomass? And it does, in the sense that, the efficient genotypes, then they might produce, in this case, they produce twice as much biomass than an inefficient one. So again, good evidence that selection and subsequent breeding could enhance the efficiency of sugarcane genotypes for, requiring less than the current fertilizer rates. We then went on and looked at this nitrate paradigm that I told you before. Nitrate is really bad news. It's mobile, it's easily degraded into nitrous oxide, and it's just not something you really want to have in soil. And yet agricultural soils generally have a very high presence of nitrate. So we thought we'd do some tests, and this is just one of many, and I apologize for those who've seen this slide before, but we basically incubated roots of young sugarcane plants, fertilized or unfertilized, and then provided them with a choice of two nitrogen sources, ammonium or nitrate. And you can see after 2 hours, in this case, the unfertilized ones, they took up more ammonium but also a fair bit of nitrate. But once they were fertilized, they had a very clear preference for ammonium. And that means that sugarcane is preferentially taking ammonium, and it's discriminating against nitrate, which would exacerbate the problem that we have. However, you might say, well, that's all, it takes up less nitrate, but what about storage? A lot of plants take up nitrate and, save it for a rainy day, so to speak. So we did test this, and we could see that nitrate storage in sugarcane, shown here, was very low. There's some nitrate stored in the root, very little in the stalk, or none in the leaves. Same with the ancestral Saccharum spontaneum. Sorry, this is a commercial sugarcane. If we look at related species like Ariensis, which is another giant grass, it's actually a bit more able to certainly store nitrate in the above ground. Sorghum and maize are, again, better than sugarcane. So we do find differences in different crops, and these are all quite close related species that differ. So that sort of then gives good indication for further selection and looking for the ability of the crosses between Ariensis and commercial sugarcane that are slowly becoming on the market because people are looking for fiber canes. They might actually have some benefits when it comes to mopping up some of this excess nitrate from the soil. Now, and I said nitrate is prevalent in crop soils and susceptible to loss, but it's not the best nitrogen source for sugarcane. So it may be different other crops, but sugarcane is not in that category. And that brings me to the sort of whole thinking of soil. So we've got these different sources: organic nitrogen, ammonium, nitrate as inorganic sources. Do we really know exactly what the plants are using? And in fact, it's an extremely difficult topic because these are all fluxes and multiple pools that occur at microsites in the rhizosphere, for instance. And so a couple of years ago, we started thinking of new ways of tackling this. And here's Richard Bracken, looks a bit like a soil surgeon, and this is exactly what he does. He has equipment that's normally used for neurosciences work, tiny probes that get inserted in the soil, and they measure fluxes. For the first time, are we able to measure fluxes? This is done by a group in Sweden, our collaborators, Torgny Nesholm and Erik Inselsbacher, who pioneered the technique. But it's a tiny probe that you insert in the soil. The flux rate, so you pump deionized water through this, exchanges with the soil solution, and you actually get an idea of what's really in the soil. And I don't want to dwell on this because this is really Richard's story, and he will give a seminar at some stage. But the bottom line of what we've been finding is that organic nitrogen that we never find in high concentrations in the soil, and that's why people always say plants use ammonium and nitrate because there's lots of it there. May not be the right way of interpreting the data, because when you look at conventional extraction, that's certainly what you do. So this is now salt extract of soil, which is conventionally done to identify what's there. So in urea fertilized soil, we have a massive concentration of ammonium, quite a lot of nitrate, not much amino acids, organic nitrogen. Organic fertilized soil, maybe not quite so much of the inorganics, the same amount of organic. And in unfertilized soil, all of these pools are the same. On the left-hand side, this is now with the microdialysis technique. You see urea fertilized, it looks a bit different. So we get different fluxes. And what I want to draw your attention to are these dotted lines. This is actually the estimated uptake capacity, the Imax of the roots. And you can see that ammonium exceeds the Imax in this case. And here they are sort of pretty much the same. But in all instances, we see that, the relative proportion of organic nitrogen in the flux is much larger than when we measure it with conventional extraction sort of techniques. So the organic nitrogen has a high flux rate and is also readily absorbed by plants or microorganisms, which is why it doesn't accumulate in the soil. But from that, we cannot deduce that it's not important. And so one of the things we're pursuing at the moment is trying to see if we supply more of the organic nitrogen, whether we can improve the overall nitrogen balance of the soil, and also using mutants that have changed uptake capacity of these organics in order to see whether that's something that breeders could also include in their selection and breeding. And if you're interested in more of this, like Chani and others, we wrote a review paper where we look at the past, present, and future of organic nutrients. So this is not to do with organic farming, but these are organic nutrients. These are nutrients that have a reduced carbon compound as part of their structure and that are probably more important than the 20th century plant nutrition researchers sort of made it out to be. At least that's what we think. But we need to do a hell lot more work, obviously, to really find good proof for this. I just want to end this particular theme on a quote. So the nitrogenous fertilizers, such as dried blood, tankage, fish scrap, blah, 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 they are, the excellent source of nitrogen removed from the soil with the greatest difficulty and so forth, whereas nitrates do not last over from one season to another. And so excessive nitrification, actual loss of soil nitrogen by leaching, and all those things that I've just talked about. And interestingly, however, you think, so this is something somebody said a few years ago. Well, actually, no. This quote was made in 1912 at a Congress of Applied Chemistry. So sadly, we haven't really moved forward much. But the interesting thing is that we're now, of course, considering a lot of the recycling of organic waste. What we consider waste is, in fact, a raw material and resource for fertilizers that becoming increasingly difficult to get, like phosphorus, or becoming very expensive, both in terms of environmental as well as economic terms, such as nitrogen. And moving on to, so I'm nearly finished my talk, but the microbes that are drivers of nutrient relations and crop productivity already mentioned that microbes are majorly involved in what goes on in the soil, how plants are supplied, what is being lost. And for the first time, is it really possible now to understand what microbes do? And I know quite a few of you are interested in it. And I'm just so glad that I'm living at this time of the, of the 21st century, and I'm actually living in Queensland where we have access to facilities to actually do this. And Mark Reagan and Phil Hugenholz are leading this research, but we're basically getting samples from the field. We're looking at amplicon sequencing, so getting taxonomic information on microbes, as well as reassembling genomes that are of microbes there to understand better the function, and then characterizing these microbes and testing what they do to the plant or to the, to the soil in terms of biogeochemical processes. And this is just a picture here from Yunkit Yu's PhD, where he could show that, for instance, these are in red are all the taxonomic units in the soil, listed from the highest abundance to the lowest, and then in blue are the ones that are enriched in the rhizosphere of sugarcane. Or root and rhizosphere, I should say. We combine those two samples. And you can see then, you go, wow, so there's some that are, many times more enriched in the rhizosphere or roots than others. And you can then go in and try to characterize these. Are these good? Are these bad? Are they neutral? And how does it impact on the way the crop's growing? And you can see this here. This is some of Chani, Pangfu, Lonyen's work, where you can then use microscopy. You can do growth tests. You can see, do these microbes, are they present in the roots? Are they inside, outside, and visualize them and then test what they do. Such as these tests, where these are just little sugarcane plantlets that are generated exenically, so they're totally controlled in some sort of jelly stuff that's obviously not soil, but it's a way of starting this kind of research at least, to then translate into real-world situation. But she found that Burkholderia australis, which was one that was isolated from sugarcane soil, and bacillus and other Burkholderia, they had quite different effects on the way they interacted with sugarcane. So this guy here had a positive effect. So the plants were much happier, grew better, accumulated nitrogen, were just sort of more vigorous. This one here, there was no effect whatsoever. and then this one was actually a baddie, so the plants didn't look very happy. So now we can actually have, with new techniques, go in and work out what exactly is it that allows these different organisms to interact differently, and how can you maybe pick one or promote a soil the way people have promoted rhizobium in soil and selected really good ones that are effective nitrogen fixers. Can we do something similar for nitrogen fixation, or in general terms like hormone production or stress resilience or other things that people find where plant growth promoting bacteria have their impact on crops? And so moving on from here to trying to sort of see, so on one hand, we can do really high-powered molecular work. On the other hand, we're still facing the issue of, how does this translate back to agronomy? And that's obviously a big stretch. And, when you think of it, I love this image that Stephen Long gave me some years ago, where, agronomists are seen as plant molecular biologists doing, really pretty menial things. And then plant molecular biology is viewed by agronomists, mostly involving Arabidopsis that is sort of dead or semi-dead. But of course, the skill is to try to bring those two extremes together. Can we do something useful with all the tools that we have that will actually help agronomy, or in this case, the nutrient relations? So ideally, what I would like, and nobody has delivered this yet, would be sugarcane that can fix nitrogen. Peter Greshov isn't here, but he's been talking about this for a while, but hasn't quite delivered yet, so maybe that's something we still see. In the absence of this kind of plant that doesn't quite exist yet, what's the second best thing? Well, that's what I want to briefly talk to you about, and that actually involves now something that was quite new for me. I mean, I did some work on farms. I like farms. I can drive a tractor. But to actually talk to farmers and try to work out, how can we string our knowledge and the societal needs together with, running and improving farms. And here you see a few people I want to introduce you. This is Richard again. I just want to point out to some strict adherence to the OH&S regulations. And this is Brian Grenshaw and Terry Grenshaw. They are farmers in the Burdekin. And this is Simon Matson, who's a sugarcane farmer in Mackay. Both Brian and Simon are Nuffield scholars, and that's actually something really big. You may not be familiar with it, but there's a foundation called the Nuffield Foundation, and they take, they're very prestigious awards where farmers apply for them, and they get to travel around the world and see farming systems and learn and get empowered to be innovative and really, promote best practices. And that's a fantastic thing. So we're very lucky that we can work with people who so deeply think about what they're doing and want to improve things. So Brian is a real character. You can probably tell that already. And Simon here also, he's very switched on, and he's got an earthworm here. So we've actually been looking at his soil and, from various angles. But the key thing I want to briefly tell you about is the nitrogen supply via legume, sugarcane or sugarcane soy, some companion cropping that we also call the Grenshaw farming system. I just heard this on a video the other day, so I'm going to use this now because Brian Grenshaw and Terry Grenshaw, they tried this for the first time. This is a picture from about 10 years ago where they successfully had an undercrop of soybean growing with their sugarcane. The soil was dying over the growth period as the sugarcane was outshading it and delivering fixed nitrogen, so moving away from synthetic fertilizer into biologically fixed fertilizer. And there are many considerations, and Brett Ferguson and others are helping to, put ideas together of what kind of legumes can we use to grow with sugarcane in terms of, being efficient nitrogen fixers, not being too competitive, being resistant to diseases, and maybe doing other things like, suppressing pathogens and having beneficial biogeochemistry that would avoid these nutrient losses that we struggle with. And really, in summary, this sort of optimizing the physiology of the plant, the microbial communities, crop management to really nail down the greenhouse gas emissions and nutrient losses, and soil degradation, which is another topic that I didn't want to weave into this, but that's been plaguing the agricultural industry and is intricately linked to these nutrient losses that we talk about. So more fertilizer is needed if the soil isn't delivering very well. But maybe a way of thinking of using less of those synthetic and mineral fertilizers that are not just, they might be supplying your plants with nutrients, but they carry the risk of losing carbon. If we lose soil organic matter, that's a big problem in many many Australian soils, but to use more organic fertilizers, including those that deliver organic carbon, and also to switch to nitrogen fixation in a larger extent to get around the need for energy in the transport of synthetic fertilizer that doesn't have a long residence time in its current form. And again, if you think of the nitrogen cycle, I mean, I sort of laugh because there's not really a cycle. I mean, yes, okay, it all stays on the planet, but, we produce it and then pump it out somewhere into the ocean or into the air. And, it's sort of 70 million tons of urea and are produced every year. And all of us actually, changes to 7 billion. It's actually 37 million tons of urea and are excreted by humans every year. And we're not recycling this, and we should be recycling this. And there are ways of recycling this, and people are starting to do this now. So farming in the future, there's going to be robots involved, that's for sure. And I mean, this probably shows my age, but when I think robots, I think R2-D2 and CP03, but there are actually other robots. And I just want to finish off with a very quick video, which I think I thought you might like. Hang on, I just quickly have to pull this up. Just finishing up, there's obviously lots of interesting things that the future holds, and I hope it's not going to be getting back to the dark ages, because sometimes when you hear what the government's talking about in terms of science funding and others, and might have to make a stand in the next few years in order to keep the ball rolling. I think that's my suspicion. But I really want to thank everybody for being here today, and I particularly want to thank Prakash, without whom this would have never happened. We would have never been able to work on sugarcane. Nicole, Richard, Chani, Thierry, Phil, Mark, Togni, and many students, many sugarcane growers, and many other people. So thank you very much.